we're back. It's 1 o'clock and we're going to be talking over the next half an hour about data omics, predicting risk and progression. And I'm joined by three brilliant researchers. Why don't you introduce yourselves? Petra, why don't you go first?
Okay, I'll go first. Hi, everybody. Adam, thank you for having us. And for me, it's the second time, so it's really nice to be back. So my name is Petra Proitsi, and I'm Alzheimer's Research UK Senior Fellow, and I'm based at the Department of Basic and Clinical Neuroscience at the IOPPN at King's, so very long.
Thank you. Christina.
Yes, for me it's the first time in the chatathon. I'm a bit nervous, and I'll just introduce myself. So my name is Christina Regido-Quiri, and I am the head of systems medicine in a hospital in Denmark, and also in the Institute of Pharmaceutical Science at the King's College London.
Thank you. Mariam, am I saying that right?
Yes, that's right. To be honest, I used to be a teacher, so I answer to anything now. My name is Mariam Shoaei. I'm a senior research fellow with John Hardy's group at UK DRI, that's Dementia Research Institute. And I've been here for the past 10 or 12 years, I would say. I work with genetic data, and I'd love to discuss that with you soon.
Well, in that case, but I stick with you because a lot of our guests will have, watchers at home will have heard of John Hardy this week, because of course he was on the news yesterday. He's been doing the rounds. Why don't you tell us about your work?
Right. Well, it's appropriate that I'm working from his office today. Then in that case, I've taken over his office today. So I've been working with John in various roles for the past 10 years or so. Most recently, in the past 6 to 7 years, I've been concentrating on using statistics and genomic data to come up with ways of predicting whether someone is likely to get Alzheimer's disease or not when they are older. And this is something that I know you focused a lot on in your chat already, using different ways of predicting whether someone is on the way to getting Alzheimer's disease or not. But I guess the major difference is that your genetics don't essentially change for most part throughout your life. You do have some rare cases where you get new mutations in your genetic material, but mostly it stays the same. And that means pretty much as soon as you're born, in theory, we should be able to have an idea of whether you will go on to develop Alzheimer's disease or not, or any other form of genetic disorder, anything that has a genetic component. And that's essentially what I'm doing, coming up with different algorithms, different predictive models that include genetics as well as other clinical factors, to see what are the possibilities we can come up with in predicting whether someone will have Alzheimer's disease or not. Yeah.
Fascinating. I'm going to come to Petra next, because I think this is all going to come together nicely for my questions.
Yes. So So I've been working on something quite similar, I suppose. So I use genetic data a lot. And we use also, I collaborate actually with Christina, so this is going to bind all very nicely, Adam, in the end. We use also other types of data, biological data, such as metabolomics or lipidomics, so different types of biological modalities. And we try to use all this together, so the genetic data, the other types of biological data, and clinical or lifestyle data to improve prediction, risk prediction, who's going to get Alzheimer's disease, can we predict it early? And also we are interested in identifying whether any of these molecules are the cause or the consequences of Alzheimer's disease. So are they contributing to the risk, or are they a result of Alzheimer's disease? And we do this using big genetic data and big biochemical data and big data sets with other information available.
And does that work both ways as well, in so much as you can also look at those kind of, what do they call them, super agers, and people to see why not only why people do, but why people don't? Is that part of that as well?
Yes. And as Mariam says, when you're born, when you're born, your genetics don't really change. So this is something, having this information later in life is actually helping us to see what are the differences in those people that they have achieved such a successful aging so far.
Fascinating. Christina. Sorry, I caught you on mute, didn't I?
Yes. So how I contribute to this omics research is that I have two scientific hats. So I studied chemistry a long time ago, and then more recently data science. So the chemistry side is producing these omics data, and to do so we have machines, and these machines are called mass spectrometers, and they're very powerful and complex machines. And so what we do with these machines is that we can get a bit of a biofluid, like very small amount of plasma or blood or CSF. And also we can analyze for biopsies, let's say like a small biopsy of a brain that was donated to a biobank. And then by using this technique, what we can detect is thousands of molecules. So these molecules could be fatty molecules, like there are many known, up to 40,000 different fatty molecules, which I think people don't know that. And also, let's say, proteins, which again, there are in the thousands. And then what we try to do by having all of this information is using data science to see how these molecules interact within themselves in the disease. And then through this, we find biology and opportunities to see how drugs can play with this biology.
So we've just, in the last session, we heard from Mark and Rich, who were talking, they very much in the lab kind of interacting with these things. So yours is off to the side of that, or does your research come first? Do you help them kind of find the right people and targets in the first place? So maybe let's ask that question first about I introduced the session as omics. Is omics just genetics then? What is omics for the people watching who don't know what omics is? Mariam, why don't you go? Why don't you take that one?
Yeah. Okay, so I'm going to put my teacher hat on. And I remember back in the day when I was a teacher at GCSE level, you would hear about you would know in GCSE biology that you had DNA that transcribed to RNA, and that went on to produce proteins. Every single one of these layers has its own omic. So if you have DNA, you're dealing with genetic data, and they call it genomics. If you're dealing with RNA, you're dealing with transcription data, you're calling that transcriptomics. And then if you go further into protein, you've got proteomics. So basically, omics is just a lazy way of saying, let's study all of that together, or parts of that together, or parts of that separately. That's the teacher way of explaining it.
No, I think that will have helped a lot of people watching, I think, because we do hear that term. So does this omics, I mean, how reliable are you, or how confident are you that we're going to be able to really understand somebody's risk? I mean, what are we from the information you have? Petra. Yeah, I'm putting you on the spot. You better all leave your mics open mute.
Yeah, you're right. Sorry, it's I'm so used to sort of like
no, I agree. And I just want to bring everybody into the chat, that's all. So how confident are you? I mean, that that this is so. I mean, I'm putting myself in this. I'm just a person too. I mean, we always hear from some people who don't want to know, some people who do want to know from their genetics as to what their risks are. I guess a lot of this depends on whether there's treatments, and if we know who's most at risk, then we have something we can do for those people. That's got to be a good reason to know this, right? So how close in your research are we to being able to predict?
Well, yeah, that's a sort of has different angles, this question.
Sorry.
I think we are no. And please, the other ones can also jump in if I've missed something. But I think we are getting close in predicting some things. For example, knowing the genetics of Alzheimer's disease, for example, we now have genetic scores where we can predict with good accuracy if someone is got higher chances of developing dementia or not. So these are quite accurate. We in my sort of like work, I'm trying to use information based on other biological layers, as I said, or for example, you're using lipids and try to see if you have higher scores, sort of higher genetic levels associated with these lipids, are you more likely to get dementia? So we are on that level, we are not there yet, but I think we are getting to a point where we can pretty much start predicting if somebody is going to get dementia. Now, how is this going to use? I think we're still in the research phase.
It's a whole separate conversation, isn't it, about use of that?
In fact, I had a debate with students debating whether they would want to know the risk of having Alzheimer's disease in the current climate, in the situation, and it's quite a big debate, I think. But I think research-wise, we are getting there. We can we are closer to predicting disease onsets, if somebody disease risk, and I think we can understand a bit more about progression. I don't know if the others have anything to add.
Yeah, have you got anything to add to that, Mariam?
I would just say that, I mean, there are studies that have looked at just genetics, which is actually a small section of the disease, but genetics by itself was able to predict whether someone will go on to get Alzheimer's disease in about 85% of the cases. So we are actually, you know, quite close to having a very good clinical predictive models using genetics data alone. But then, as you say, that leads on to the question, do I want to know that right now anyway?
What do we do with that information?
Yeah.
And this session's talking about progression. So this same information you're looking at can not only tell the likelihood of getting it, but then what will happen to you after you get that. Is there anybody, I don't know, is that something you look at, Christine? It sounds like you're on that.
So are you referring to conversion or?
Well, maybe it's more one for Mariam about disease progression. So, you know, we know that everybody experiences symptoms differently. That you know, we talked to Chris this morning that has mixed dementia, who has changed some of his lifestyles, but has been living with the disease for a number of years now. Whereas we also hear from people who very rapidly progress. Is the information, the research you're doing, able to help with that?
Well, that's certainly the hope. That's what we are aiming for. We know that dementia progresses. Everyone experiences it differently. So there are people who are diagnosed, and within 5 to 7 years, they've completed their course and the disease is over. But some people go on living with dementia for 20 years, and for the first 5 to 7 years are actually relatively, you know, fine. They, you know, we heard this morning about Barbara Windsor's experience, who was actually able to hide the symptoms for quite a long time, it seems, which means that she wasn't progressing that rapidly from the beginning. So what dictates whether someone goes very quickly or very slowly must be genetics as well as environmental. So environmental is a little bit more difficult to understand because there are so many different factors contributing to it. But at the very least, we could try to identify what are the genetic risk factors that help push someone towards a faster progression as opposed to a slower progression. And we've done that with other disorders. We've done that with Parkinson's disease. We've done that with progressive supranuclear palsy. It's just been released that there's a study that's looked at multiple sclerosis, where they've actually identified genetic factors that push you towards faster progression. And in 2 of those, the first 2 disorders that I've mentioned, that's actually led to pharmaceutical companies looking at targets, molecules that they could actually aim for to reduce the speed with which people progress, those who do progress.
And that's, I think, what you're looking at, Chrissy, isn't it? Is replenishing those molecules.
Yes, so, because we combine genetics with downstream products of the genetics. So since we are also looking at the proteins and lipids, then we can we can start looking at the ways of modifying the disease. So talking about the metabolic risks that might affect also progression to dementia, as we know, like diabetes is a risk for dementia as well. So all of these environmental factors are going to be seen in the phenotype, in the proteins and the small molecules.
And I mean, how do you practically replenish a molecule? Is this taking a tablet? Is this something you can do naturally through a change of lifestyle? Because we said you can't really change your genetics, but how can you change the molecules that are there? Who wants to take that?
you can do both.
You can do both.
Yes. Yeah, you can do both. You can have molecules through nutrition, obviously, like we all know about the omega fatty acids, and how we need for them to enter the brain, and actually they enter the brain attached to bigger lipids. So these are the kind of things that we discovered in our group, and of course the genetics people can comment on how you would also need the drugs as well, right? So
go on, Petra.
No, I agree with Christina on that. Yes, it's through diet, for example, but also lots of proteins are drug targets as well. So by identifying a protein that may be associated with Alzheimer's disease, then you can see whether this is a target for a drug, and then we can start intervening. So it can be used both preventative as sort of changing your diet, for example, or as an interventional approach.
So at the moment, do you work on kind of big data sets, or are we talking about individual people? Because when we're talking about the lifestyles or potentially replenishing molecules, of course, you can only do that if you're then going to have somebody to work with to go away and work with on this. Or do you work with big data sets, or do you do both?
Big data sets of many people. Yeah.
What about you, Maren?
Similarly, I mean, when you're dealing with genetic data, you're dealing with millions and millions of variables that you're looking at, and at the same time, because I mean, for anyone who would do statistic will know that you cannot look at millions and millions of different things in 20 people, in 40 people. It has to be closer to 40,000 people to be able to actually reliably analyze your data. So big data is definitely, I guess, a common theme here.
And so that's where I guess we need more people to come forward and contribute to things like the genome studies and the biobanks and the different trials that are taking place.
Yeah, absolutely.
Are there any that you'd particularly like to get a plug in for? I'm also, we should talk about things like Dementia's Platform UK, which are amazing tools funded by the Medical Research Council, I think is in the UK, that funds that, which is this kind of collaboration of different cohorts of data from different places that puts it all in the hands of researchers, mostly for free, I think, to perform exactly what I think we're going to talk to some people later on today talking about drug repurposing, for example, that use exactly those kind of data sets. So do King's and UCL have their own, have you got your own amazing data sets to do this work on? Not that I'm introducing an element of competition.
We don't share.
I'm sure you do share. We do. Everybody shares. Do you have your own data sets? I mean, so that's a question. How do you get your data to do this work on? Where does it come from?
I can answer part of that, at least from my side. So a lot of the work that we do is heavily dependent on the data that's publicly available. most of those are academic collaborations that have made it into the public domain. So for example, there's something called the Alzheimer's Disease Neuroimaging Initiative, which is an amazing data set where they look at people who are healthy as well as people who are cognitively impaired and analyze different aspects of their life as well as their brain, their genetics, etc. There are many data sets. For example, the UK Biobank data, which is one of the most powerful one. In fact, if you go to a genetic conference now, you can bet that at least a third of the people will be using the UK Biobank data set to do anything. It's absolutely amazing data set, and we need more fundings for initiatives like that to be spread across Europe and across the world. And I hope one day we can see that in other populations, not just the European population.
And this is something that many people who are watching might be involved in this and not necessarily entirely realize, because when you interact with healthcare, next time you're asked to fill in a questionnaire, it might say, oh, can we just use this little bit of information we've collected for you and contribute towards these cohorts of data, which then feed into these bigger databases that are they're all there. Conspiracy theorists might not like that idea, but it's there, and it's making a real difference, because without you and volunteering this information, we wouldn't have the data to do the research on. What have you found that surprised you so far? There's something kind of, I'm going to make this a two-part question. What have you found anything that surprised you, and have you made any changes to your own lives as a result of your own research? Christina, you go first.
I have found a lot of things that surprise me when I do research. Let's see, like some of the things that surprised me the most, because they were going a bit against the paradigms. When we had a look at the brains from people with Alzheimer's, we actually found that the molecule of cholesterol was decreasing, both in the brain and in the CSF. And like everybody thinks cholesterol is bad for you, but in fact the molecule of cholesterol is doing a lot of roles in the brain, and it needs to be in the right place. So the problem is when it doesn't go where it needs to go. So that was one of the results that surprised me. And then another thing was to do with what I was hearing in one of your interviews this morning at 9, I think, and it's to do with sleep. So we also found that the people with Alzheimer's have molecules in the blood that are increased and are to do with the lack of sleep. So, and that was at the really early stages of Alzheimer's. So I think this is a bit related to maybe changes that I have done in my lifestyle as well, which is making sure that.
Have you just given us all permission to it's okay to have high cholesterol? Did I get the wrong takeaway from that?
No. This is to do with sleep.
No, I joke, of course. But so that's fascinating. I mean, it's great that, again, it kind of adds to that thing about collaborations and why the DRI and other organizations that work together are so great, because, you take what you learn about cholesterol and then find somebody else who's working on cholesterol somewhere else that is connected, and it's brilliant. What about you, Petra?
I haven't found anything that surprised me so much. I came in with no major expectations, or at some point I realized I should not have major expectations from the data we're getting. What has surprised me a bit, which now it doesn't anymore, is that using these methods we're using, we found a lot of associations, we thought, between some molecules and Alzheimer's were the other way around. For example, we thought that, okay, let's say cholesterol, although it's just an example, is associated with Alzheimer's, maybe it's contributing to the risk. And then we thought, actually, it's not. It's maybe because you've got Alzheimer's disease, you've got higher molecules. So we started sort of disentangling some of these causal associations, and some of them were the opposite to what we expected.
And that's where your data can come in again, right? Because you take data from people at different stages in their lives. So you can look at this, you've 40 1-year-olds and then your 42s and your 43, and you can build up that bigger picture.
Yes, you can do it with different types of data and different types of approaches. And some of this data doesn't even need sort of longitudinal data, some of these approaches, but it's always nice to sort of, yes, triangulate this information, put it together. But
What
yes,
about No, that's all right. And Mariam, have you changed your life? Have you found something that discovered you? I mean, you're working for the famous John Hardy. Surely you're making new discoveries literally on a daily basis.
Well, I have to say, I came into this field from a background. My PhD is actually in physical chemistry. So everything was absolutely new to me and everything was surprising to me. But over the past few years, I found that so I've actually really enjoyed how things click together. I was like, oh, of course it does. Of course, that makes absolute sense. But what really did surprise me was actually looking at progression. We've realized that the genetic factors that drive risk in Alzheimer's disease are not the same ones that drive progression at all, which means that you cannot be targeting the same molecules if you want to slow down the disease. And I found that it was one of those, how could I have possibly thought otherwise moments, but also at the same time, oh my God, that's really amazing type of moments.
It makes complete sense, doesn't it? I mean, I can see how you jump to that conclusion that the same things that cause it are the things that affect progression, but to disassociate those two things, I think you're right. When you say it now afterwards, it makes complete sense, but it doesn't at the time. I'm afraid that's all we've got time for. Fascinating as ever. So I'm joined by the wonderful Christina, Petra, Mariam. You'll find details on them and their work and links to their Twitters. I think you all have Twitter, you're all Twitter users, all on the guests page of the website. Thank you ever so much.
Thank you so much, Adam. Thank you.
Please do keep up the amazing work, and I hope we'll get you all onto Dementia Research to do a podcast on this, because it's a fascinating topic that we've barely ever touched on in the podcast. Thank you very much. Have a great day.
Thank you so much. Bye.
Everyone.
Okay, so we've hit 1:30, so we're 5.5 hours into our 12.5-hour live stream, only half an hour away from the halfway mark, and coming up between 1:30 and 2 p.m. we've got a session which has been given the title Doing Clever Things with MRI, which I think is a bit of a broad title because I gave it that title probably before reading everybody's bios and realizing they do far more than just do stuff with MRI. They do all kinds of clever things with lots of stuff. Just a reminder before we move into this next session, all of our brilliant guests are here. We've got Ricardo Manca, Jodie Watt, and Dr. Claire Lancaster. I should call you all Doctor goes before all of those things. Who have come from, you're all from the UK this time around, so we're going to move on to this session now. Just a reminder to everybody that not only are we sharing information about research today, but we are also hoping that you'll very generously give whatever you can to our amazing 4 charities who we're here to support. We have the Lewy Body Society, Race Against Dementia, Alzheimer's Research UK, and Alzheimer's Society. Of course, there are many more that we could support, and we have lists of those on the website as well. If you go to chatterthon.uk/donate, or go to the chatterthon.uk website, you'll find a link there on how to donate. And please do continue to give what you can. Our funding target this year was probably a bit ambitious, but honestly, everything you give will be equally divided amongst those charities, and it'll make a difference. So before we just move on to this next session, I'm going to play a quick film from who we're going to go to this time. I think this time around we're going to go to one about the world, about football, because it's the World Cup. So we've got a video about the World Cup and Alzheimer's Society.
What time should we leave tomorrow?
I think about 8. What do you reckon?
Yeah, that sounds about right. What time should we leave tomorrow?
I told you, 8.
What time should we leave tomorrow?
We're leaving at 8, Jane.
time should we leave tomorrow?
I think we should leave at 8.